Hi, my name is Thatchayut Unjitwattana. I am a graduate student in biomedical engineering from the University of Michigan. Today, I'm going to talk about the single-cell pathway embedding neural network, or scPENN, for single-cell RNA-seq analysis. Deep neural network models are more potent than the traditional machine learning approach in classifying multiple cell types in single-cell RNA sequencing data, since it captures features and characteristics of high-dimensional data better. However, pathway embedding as a form of enhancing interpretability of neural network is currently lacking in the single-cell RNA-seq analysis domain. Here, we introduce a single-cell pathway embedding neural network, or scPENN, which integrates the gene-pathway relationship at the connection between nodes in the model. The connection between the gene layer and the pathway layer is determined by the adjacency metric, in which the Aij is equal to 1 only if a gene I is a member gene of the pathway J. Otherwise, Aij is equal to 0 as shown in the equation 1. The rest of the connections are fully connected. The pathway score of each pathway could be retrieved by extracting the output of node in the pathway layer. This pathway score is calculated as shown in the equation 2. In this study, we used the dataset GSE161529 containing single-cell RNA-seq profiling of breast cancer tumors, BRCA1 mutant preneoplastic mammary gland cells, and normal mammary gland cells. We selected samples from 3 patterns of different categories to train the model, including total cell population from a normal pattern, from a pattern with BRCA1 preneoplastic, and from a pattern with triple-negative breast cancer. We annotated cell types using the single R with the Human Primary Cell Atlas as the reference dataset. Then we manually selected 11 annotated cell types for the training dataset, including B cell, chondrocyte, dendritic cell, endothelial cells, epithelial cell, fibroblast, macrophage, monocyte, natural killer cell or NK cell, T cell, and tissue stem cell. To define the connection between the gene and pathway layers, we selected 10 to 75 pathways from 3 categories in MSeqDB database, including 186 pathways from the KEK subset of canonical pathways, 189 pathways from oncogenic signature gene sets, and 700 cell type signature gene sets. We compared the performance of scPENN with one machine learning model and 2 deep neural network models, including random forest, the fully connected deep neural network, or FNN, and the graph-embedded deep feedforward network, or GDFN. GDFN is the deep neural network that integrates external relational information of features into the deep neural network architecture. The sparse connectivity in GDFN is determined by the gene network from the HIN database, which is a database of high-quality protein-protein interactomes. Since the sparse connectivities in the scPENN and GDFN are defined based on different biological prior knowledge, they can be viewed as different implementations and also the specification of the neural network models. As you can see in the table 1, scPENN can perform better than benchmarking methods in almost all metrics on the internal validation set. While there is a trend of increase in majority of metrics when comparing scPENN to the FNN model, the difference is not statistically significant, and this might be due to the high similarity between the training and whole dataset. For that reason, we introduced additional external validation datasets in which samples are different from the 3 patterns in the training dataset. We observed that scPENN yielded statistically significant increase in performance in almost all metrics compared to the benchmarking method on the external validation set, as you can see in the table 2. Although there is no significant increase in MCC in scPENN compared to the random forest, its mean MCC is nevertheless better. This shows that integrating prior biological knowledge into the neural network model could potentially make it more predictable in new patient samples. To observe the interpretability of the model, we extracted pathway score of 1075 pathways from all samples and then generated the UMAP plot. The UMAP clusters show high degrees of correspondence with the cell type annotations by predefined gene markers, as you can see in the figure 2a. Moreover, when compared with the UMAP plot based on top 1075 viral genes, the advantage of the pathway approach is apparent. With pathway-based UMAP clustering, the epithelial cells and tissue stem cells, which form epithelial cells in the differentiation, are closer to each other as expected. Whereas the gene-based UMAP, as you see, tissue stem cells are wrongfully closer to endothelial cells. This demonstrates that pathway scores preserve cell type identity as well. We then selected the top 20 pathways according to their variances and show the right clustering results on them, as you can see in the figure 2c. Cell types with similar origins are clustered together. Immune cell types, including T cells, NK cell, monocyte, dendritic cells, and macrophage are in the cluster, whereas epithelial cells, endothelial cells, chondrocytes, fibroblasts, and tissue stem cells are in the other cluster. Since KEK pathways have well-documented and curated biological functions, we extracted the 186K pathways from the 1025 pathways. Interestingly, T-cell receptor signaling pathways has a significantly higher average pathway score in the T-cell population, as you can see in the figure 2D. The specificity of the T-cell receptor signaling pathway scores in the T-cell population resembles that of the CD3D expression, which is a known T-cell marker gene in the T-cell receptor CD3 complex involved in T-cell development and signal transduction. This suggests that this pathway could also serve as T-cell specific marker as well. Other than that, Chavelu is used to measure the importance of each input feature to the prediction of each output class, providing another aspect of interpretation to the neural network model. We then verify top genes via the literature review. We found that each top genes play an important role in the functions of each specific cell type. For example, CXCL8 has the highest Chavelu for the epithelial cells. This is one of the biomarkers for the epithelial cells and acts as a multifunctional cytokine to modulate proliferation, invasion, and migration of tumor cells by an autocrine and paracrine. This gene also has the highest CHA values in the tissue stem cells, as it is involved in the maintenance of cancer stem cells. In summary, we propose a new model called scPENN that utilizes the biological prior knowledge to construct connections between the genes in the input neurons and the first data layer of neurons denoting pathway. The gene-pathway relationship can be automatically configured in the neural network model based on the agency metrics provided to the model. This makes scPENN scalable and adaptable as knowledge in databases of pathways grow over time. By the way, other prior knowledge could also be integrated into the model as long as the agency metric is created to describe the relationship between nodes in the input and the first data layer. Thank you for listening.